Should we start or wait?
Just 2 seconds.
Sure.
So you can start.
Okay. Okay, welcome back everyone. Another lecture, but it will be the last one for today. We are continuing to discuss stochasticity. I hope you're still surviving.
Yeah.
We're trying to, so again, what we're doing, I'm giving you different examples of the application of stochastic approaches, and with additional background to some specific processes, so you could see how important is stochasticity. So let's go to another story, and this is biological error correction, molecular mechanism of biological error correction. I encourage you again, ask me questions, interrupt me anytime. So we already mentioned a little bit that biological processes exhibit remarkable accuracy. I give you different examples, but I could also give you this example that in DNA replication, when two cells divide, they have to exactly replicate DNA, and the errors are not acceptable. So the error rate, the probability of error is very low, 10 to the minus 8, 10 to the minus 10. Okay. So the typical size of human genome is 10 to the 9. So it's essentially less than 1 error per genome can happen. If you look at other biological processes like RNA transcription, the ones that we mentioned in previous lectures, error rates are maybe not as impressive, but still pretty good. Error is 10 to the minus 4, 10 to the minus 5. Or if you go protein translation is synthesis of protein synthesis. The error is also not as good as here, but still quite low. This is the probability of error. Why I'm saying that this is actually surprising? The reason is that, let's come back to the example of DNA replication, right? So DNA is actually, it's a heteropolymer consisting of 4 different types of nucleotide: cytosine, guanine, thymine, adenine, and the picture shows you that they're chemically very similar. They're not exactly the same, but very, very similar. So in other words, the wrong one can attach and can be more or less stable. So the difference between right versus wrong comes in order of 1 hydrogen bond, which is several kT units. If you think that this process of coming wrong and binding is followed by equilibrium, that would be the error. So this is the smallest possible error if the process is equilibrium. But the error that I showed you, error rate, the probability is much lower, which is indication it's non-equilibrium process. We know all biological process non-equilibrium. So the ability to sustain low errors in biological systems is consequence of non-equilibrium process. So another things to remember, cell environment is a very mixed environment. So many mistakes are possible. So biological error correction is needed if you want to have a robust outcome. Now, there are 2 important people who did contributions into this kinetic proofreading, and one of them is John Hopfield, Nobel Prize winner in physics in 2024. Not for this work, for associative memory, although I think this work is better. Another person was Jacques Mignot, French scientist. So the Hopfield had a paper in PNAS in English. Jacques Mignot had a paper in French in Biochimie. Who reads French journals? I don't know. So people don't know much about Jacques Mignot, although the work as good as the Hopfield work. So they come out with this idea, which is known as the kinetic proofreading. And the idea is the following one. Imagine, so this is the scheme that we were discussing with the Michaelis-Menten scheme. So there is an enzyme, there is a substrate comes, binds to enzyme, and then essentially make a product. Process without this kinetic proofreading. But the idea of Hopfield and Meunier was that there is additional step, and from this step, system can reset. Somebody asked me about stochastic resetting. That's an example. And that's the way to correct it. If you put a wrong substrate here, this is the way you can correct it. This is the possibility of correcting. So this kinetic proofreading, EPR, I'm sorry, I'm using these abbreviations. It's a kinetic proofreading. It's a non-equilibrium process that utilizes this free energy difference. So it uses free energy difference between right versus wrong substrate to do this work here. Let me give you an example so you will understand. I was thinking about the example. Let me give you an example. So, do you know what is this painting? Huh? Van Gogh. You're a very educated person. Van Gogh. This is one of the most famous paintings of Van Gogh irises. So imagine that somebody brought Van Gogh to IIT Rapar, and let's say students can go and see Van Gogh. And let's say 50% of students like Van Gogh, 50% of students do not like Van Gogh. And administration said the rule is, if you like Van Gogh, you can stay 10 minutes in the room where is this painting. If you don't like it, 1 minute. So the blue people like Van Gogh, orange do not like. And so, and you have room people enter, exit, enter, exit. You agree with me that if you continue this, at some point, you will have bigger fraction of Van Gogh fans inside, right? So the point here is, despite the fact that in outside students world of IIT Rapar 50-50, 1-to-1, inside of this room, you have significant enrichment of those people who like Van Gogh. That's an analogy of kinetic proofreading. Okay, a little bit elaborate analogy, but that's an analogy. You are using this free energy difference in this language, liking or not liking, to improve, to increase the fraction of the right outcome. In this case, those people who like Van Gogh is the right outcome. Makes sense, right? Okay, so this kinetic proofreading enhances accuracy by resetting the system to its initial configuration, and that's what allows the system to correct the mistake. But at the same time, if you think a little bit, yeah, this is good, but biological processes should happen also fast. You cannot forever try to correct mistakes, right? When you probably do writing in some essay in Hindi or English, right, in your school, right? Then you go and check it, but you cannot do it forever, right? You have to, at some point, give it, right? Because there is exam has a limited time. Exactly here, you cannot constantly try to improve. So which actually means that there is a problem here which called speed accuracy trade-off. And this is a very common problem in all biological processes. There is always a dilemma to do things faster or to do things more precise. Exactly, right. And again, high accuracy, slow speed, low accuracy, high speed. And we know this if we fly, right? If you want to go soon through security, will be not so secure, but if you want to be more secure, it will be slow. Okay, so kinetic proofreading is really connected to this speed accuracy trade-off. Okay, there are problems. There are many problems with the kinetic proofreading. People still don't understand at what conditions you might achieve the best possible minimal error. Hopfield suggested one thing, which actually was wrong. Experimental suggested other things. There are other problems with the kinetic proofreading. I mentioned them here, but the most important is this one. If you open textbooks or you talk to biological colleagues and saying, they will tell you the biological systems are optimized to have the lowest possible error, right? That's what they think. It will turn out to be wrong. It's not. So the people believe the biological system trying to be as precise as possible. They're precise. Don't get me wrong, but not the. But there is a way to make them precise even more. But evolution didn't want to do it, and we will explain why. Okay, good. So we come out with a model with a stochastic approach. So you can describe chemical kinetic schemes of kinetic proofreading. And again, you use first passage calculation. Here you can do first passage calculation. And so in the simplest case, imagine this is enzyme and this is one pathway when you produce the right product, and this is wrong pathway, you produce wrong product, and or product production can be from this intermediate state. And then essentially the error rate in this language is the probability to go over the wrong pathway or the right pathway, and you can calculate the times here. Again, another example where a first passage is more convenient, but some of the theoretical work were also done in a normal forward master equation. And so we decided to look at several systems, but before again, you have essentially two identical pathways. The only difference this way or this way or this way or this way. The only difference between them is there is rates in the wrong direction would differ from rate in right direction, and we can quantify it by introducing this discrimination factor. Okay, good. So then we use this approach to consider two specific examples, two specific systems. So the first system is DNA replication in a so-called T7 virus. And the reason it's convenient because all rates were measured, right rates, wrong rates were measured. So you could go and this is quite well described by this scheme. Again, so this is enzyme, one intermediate state, second intermediate state, that come back, but from this one you can make the product here. And that's for this one, for this T7 DNA polymerase. And all rates are known. That's what I said. There is all rates are known in right direction, the wrong direction. So that's a convenient way of applying the theory. Another example is a tRNA selection, and this is in ribosome. In the ribosome, and this follows this chemical kinetic scheme. Again, all rates are known, given over here. All rates which can be which follows the schemes can be described. Okay, so we have a stochastic model of kinetic proofreading for two different systems, and this of course depends on many rates. So to understand how it works, you can keep all the parameters and vary one of the rates. That's what schematically shows. Suppose I keep everything else, so suppose I can vary one of the rates just to see how the system is optimal. And you can do it, and so this is the error as a function of one of the rates called polymerization rate. This is mean first passage time here. And again, so the red points corresponds to this minimal time, minimum error, and the same is here. It's not convenient to look at two graphs, so you can combine it in one graph. So this is the same, one graph is the same as the graph over there. And so this is mean first passage time, this is error, and this curve corresponds to different values of one of the rates, polymerization rate. And then let's look at this branch. So this branch is the one where speed accuracy trade-off is followed. So you see, if you want to be more precise, you go along this line, you will be slower because time goes up. Okay, if you want to be less precise, you will be faster. So the speed accuracy trade-off works here. But surprisingly, there is a range of parameters where it doesn't work. There is a range of parameters where there is no speed accuracy trade-off. You want to be more precise and you can do things faster. The green point corresponds to experimental data. So for this system, experimental data are on this branch. But important first conclusion is that speed accuracy not necessarily always observed. You can change other rates and situation is similar. So what is another interesting observation here is, so this is your experimental point, this green one. The red triangle corresponds to the most precise system. You see, your system that is not here. Please, let's log scale. You can get much better precision, but evolution did not optimize the system to make it very precise. It actually wanted to make it quite fast, low time, but not precise, because if you be very precise, you will be slow here. You can change some other rates, and situation is similar to the green experimental point. This is local minimum in time, which is the fastest possible position here. And then if you look at the graph, you can say, okay, good. So the systems are quite fast, experimental one, but why I'm not trying to move my system? Why evolution didn't move the system in this direction where you still could be fast and simultaneously will be also precise? And the possible explanation we will come back to this is probably energy consumption. Okay, so it seems that speed appears to be more important than the accuracy as long as the level of error is tolerable. So speed seems to be more important as long as the level of accuracy is tolerable. So that's what it is here. Let's go to the second example. So remember, we're doing two systems. We're looking at two examples. The first example is a T7 DNA polymerase. Second example is tRNA translation, which is described by this scheme. We repeat the same calculations here. That actually, this is what was done for ribosome for E. coli bacteria. And this system is actually more interesting even because not only there is a wild type, so the black curves correspond to wild type, but there are very high quality chemical kinetic measurements for mutants. One of the mutants is hyper accurate mutant. This is the purple here, and the blue one is error prone. So there is a mutant of E. coli bacteria where it can reach a better precision. That's what this hyper accurate mutation, and error is more error prone. Okay, so and even here, as you can see, so the green point is experimental measurements for wild type. This is experimental measurements for this mutant, experimental measurements for this mutant. So it seems in all cases, systems prefer fast speed, the smallest possible time, not the most precise. Actually, this is the most precise location here, where the error is smallest one. Okay, so and by the way, wild type ribosome is the fastest one. You see, the wild type is the fastest one. So, which means that evolution for E. coli bacteria, evolution probably tried all possible combinations, and evolution decided that it's better to have E. coli bacteria which do things fast enough, not precisely enough. Okay, which is interesting result. So nature didn't choose the most precise one, chose the fastest one, as long as the error is tolerable. So that's what it is here. So it seems the tRNA selection by E. coli ribosome is inclined to be faster with higher but tolerable error. And again, we plot, we change it in other rate, and mean first passage time, error. This is experimental observation. This is the maximal possible speed here. And the question comes, why the system is not trying to move in this direction where you can kill one bird with two stones, right? So you can be both precise and fast. And the reason is energy. You need to actually have to spend some energy. And what we did here, what we tried to mimic this energy consumption. We introduced a function called cost, and this is the ratio of the fluxes which go over loop over the total production. So let's look at the scheme. You will understand. So I have a flux of a system and I produce something, right? But if I go over the loop, it's futile. It's not productive, right? It's useless. I'm not producing anything. So the ratio of two fluxes which over the loop over the fluxes that are production is a cost, right? That's how much it costs. You do a lot of things, but only few things are useful for you for production. So that's what we plotted over here. There is a cost. So to go from this point to this point in this unit of cost, you really need to increase significantly the cost. So maybe nature doesn't want to spend this energy for doing this. It's enough to have a tolerable error as soon as the system is fast. That's what is the result of this argument. Okay. So, but the important thing here is this is stochastic approach, right? You still do stochastic model. It's more elaborate model. You take into account different states, but it's still stochastic approach. And the stochastic approach gives us several interesting results. So it seems like evolution optimized biological enzyme to achieve the optimal function of cells, and that speed is more important. But you have to be careful, right? In my previous lectures, I gave you examples that people can really come out with their own conclusion. So you have to be careful, critical. So my analysis, what I showed you, was based on two processes, DNA replication and protein translation. It was only done for bacteria, for E. coli bacteria. It's possible for more complex organisms, some other criteria might work. But at least for bacteria, it seems like that this is the case. Yeah, this is a picture that you don't need to remember all mathematics, but this is the story, in my opinion. So it seems that nature works in economic way. So this works like a car factory in China, not car factory in Germany. As long as the quality is tolerable, faster, faster, faster. While in Germany, quality is more important. That's why this stuff is so expensive. This stuff is relatively cheap. Do you have a lot of Chinese car in India or not? No. Okay, soon you will. America do not have, but this is very expensive. So it seems like a like theory of work in this way. As long as the quality is tolerable, okay? So there is no point for biological system to have a perfect precision because it's a dynamic system. Dynamic system, you cannot constantly try to improve things, right? As long as it seems to be okay, go ahead. This is the first prediction from this stochastic approach. Then second message. First of all, it is possible that this, I told you about speed and accuracy, right? And I was arguing that they oppose each other, but I give you a couple examples here. They're not necessarily opposed to each other. It's not necessary that if you need to be something a better quality, you need to take it longer time. And my proofs here come from this graph. So remember, this graph, it's a mean first passage time versus error, okay? And all we need to look at these 2 points. So that was experimental point for wild type E. coli bacteria. This is for one of the mutants which are more error prone, okay? See, this does not follow speed accuracy trade-off. This is less precise and it's slower. So it's not necessarily the case that something which is more precise need to take longer time. Oops. Good, right? So let's come to the next message. Again, BMW factory in some Chinese car factory is a good example. Energy cost of proofreading might also be important, okay? So as long as you do it fast enough with tolerable accuracy, as long as you don't pay much salary, right? That's okay. That's how nature wants to work. If you think about cost as a money. Here you really have to pay a lot of money to this guy. Okay, in Munich. I was in this factory in Munich. It's an amazing factory, but I don't think people work so long. I think 4-6 hours they work, and salary they have is like almost American professor. Anyway, so which means that eventually the world will be dominated by Chinese products at some point, unless India will start to produce the same way. And that's also the reason why there is a lot of things made in India and all over the world. It's cheap, fast, reasonable quality. Okay. So it seems that the natural system, it seems to me, based on these stochastic analysis, work this way. So I keep telling the word tolerable quality, right? The quality is tolerable, right? So do you want your iPhone to be working for 3 years or 1 year, right? What is it tolerable? For some people, 1 year is enough. They don't want to change something. But for some people, you need 10 years, right? If you talk to your grandmother or grandparent, they will tell you, oh, they had some boots for generation, right? It doesn't impress you, but that's what they think is a good one. Anyway, so what is the tolerable level of error? It's not clear, but we can think a little bit. So, for example, bacteria genome size is small at 10 to the 6 bases, and the error rate is 10 to the minus 6, 10 to the minus 7 in replicating bacteria DNA. In eukaryotes, and humans are eukaryotes, 10 to the 9 error, 10 to the minus 6. So it seems that nature decided to do the following thing: as long as there is no more than one mutation per genome, that's okay. Why it decided this way, I don't understand, but that's a tolerable level. They decided it probably takes too much resources to keep it even below. But as long as there's no more than one mutation per genome, that's how it works. Yes.
One thing I didn't catch is here in the DNA replication when you say error, so AT and GC.
Yeah. But you can do AG, AC.
Oh, that happens.
Yeah. Okay. So this is a good question. Okay, let's come back to this. This is an important thing because they're chemically very similar. Just a second. Let's soluble, but they're chemically very similar. That's an important issue here. So you see, they're chemically very similar. I think this is the right connection. This is the right connection. But this molecule can also combine here. There will be two hydrogen bonds. These can connect with this one. These can connect with this one. They're chemically very similar. This is the biggest problem in biological systems. The cytoplasm is not a very clean medium, in the sense that there is a mixture of chemical compounds which are chemically very close. They're slightly different, but close.
So that means in the genome there are occasional.
Well, okay, we will talk about this in one of my lectures. Yes, that happens. But biological system knows this, and there is so-called DNA repair pathway. They constantly repair this. They constantly improve, because wrong connection leads to mutation, and mutation leads to diseases, cancer, and so on.
And also, when you said error rate is 10 to the minus 8 or 10 to the minus 9, that means in a string of symbols, about
one mistake. Eventually, one mistake. Eventually, after all this repair, cleaning, whatever happens, one mistake, right? It's like you writing essay, and in essay which has 10 to the 9 words, only one mistake somewhere, which is pretty impressive. Okay, so everyone understands this point, right? These compounds are very similar chemically, so mistakes are possible. There is a repair pathway, fix it, many of them, but not everything.
So how the first person time calculation rate, for example, here, when we are optimizing the parameters, so how the first person time calculation is helping in the optimizing the parameters, the weight parameters?
So you're calculating for passage, you're calculating what is the first time to go from initial state to the product, or to this one to this one. And what is convenient actually in this language, you can define this error as a ratio of splitting probability. But again, this is the way we did it. It's not, there's exactly the same way you can do it with a normal one. And then will be the ratio of fluxes in right way versus wrong way versus right way can be the error. Okay? Okay, so we go back to what is the tolerable level of accuracy. That's my politically incorrect examples. Yes. But there's one more additional thing. So what I keep telling you something that it's not good to have errors, right? I keep telling it's not good to have errors for biological systems because mutations will come eventually, might be cancer. I'm not exactly correct here. There are the systems where having errors is a good one. Viruses can survive only if they mutate constantly. That's the only reason every year we have. Do you have flu vaccinations in India too?
Yes.
Right? The reason we have flu vaccinations that the flu species this year different from what last year, from the year before. Okay, so that's why they're very highly. That's the way to survive for them. Otherwise, they would be eliminated. So what is shown here? This is genome size, and this is mutation rate. Can you guess what is the most fastly mutated virus rate here? Can you guess? HIV. No, coronavirus, forget about this. Is artificial thing. So it's HIV. So by the way, from all viruses, the most dangerous is viruses which are RNA viruses with a reverse transcription. DNA viruses. Okay, DNA viruses are those viruses where a piece of genome, piece of DNA. They're not a problem usually because DNA replication has very low error. RNA viruses has instead of DNA is a piece of RNA. And the error rate in RNA is smaller, bigger error, and that's the dangerous one. And then comes viruses which, on top of this, they have also called reverse transcription. That's those are RT, and this is HIV. Okay.
So the DNA virus, did you say it kind of latches onto a segment of the DNA?
DNA virus has its own genome, but it's DNA.
Okay.
When it's DNA genome, it's not so big problem because DNA replication has very low error.
But what does it do? Does it insert itself to the host?
It inserts itself, right? Yes, right. The most dangerous for us, for humans, for other things, are those RNA as well as reverse transcriptase. That's why HIV, flu, and so on. But important thing here is too low errors are not good for them. They need to have really changing quickly, right? If you want to attract opposite sex, we need to change, otherwise it will be boring. Something like this. I don't know how to explain it, but viruses. So and the point here is very low error is not necessarily good all the time. And by the way, evolution, the way evolution happened was mistakes were happening, and some of those mistakes were advantageous and were picked up. So if without mistakes would be no evolution, we would be no humans. Something like this. Okay. Yes, so this is the end of the story, which I wanted to. This is not a trivial story, but it's again the story where stochastic approach can explain many things. You can get a lot of things. You can again, the important thing is that using stochastic analysis, you can understand something about biology, I think, better. You can understand how the error correction works. And we apply this for two specific systems, DNA replication and tRNA selection. And we argue that those bacterial systems, biological, it seems to be they prefer the speed over the accuracy. As long as the errors are tolerable, we try to discuss what is the tolerable. It's not clear, but I was trying to argue the rule of thumb that there is no more than one mutation per genome seems to be for biological systems a reasonable rate of mutations. And then we also argue that speed accuracy trade-off is not always necessarily observed. And then there is additional things that you need to take into account, the energy, right? And if you forget about everything I told you, BMW and Chinese car producing car, right? That's probably the nature works more like Chinese car, not like BMW. That's why Europe will go down soon. Okay. So any questions? Okay.
In one of the earliest slides you had about errors in DNA, error rate in DNA, error rate in RNA, and error rate in protein. So when you say error rate in protein, does that mean that the wrong protein gets made because some?
Yeah, and that's actually, let's come back. Yeah, okay, so come back here. So let's question to you. Why do you think, it's related to what you're saying, in a moment I will rephrase it. Why do you think the smallest possible error in DNA replication, but then error is not as low in transcription of protein synthesis? What do you think? Why do you think the lowest possible error should be DNA replication, but not in synthesis of DNA? What do you think?
DNA makes the RNA, RNA makes the protein.
Okay, so yes, so protein translation is in making proteins. Why it's acceptable to have error rate in making proteins significantly larger than in making in DNA replication? What do you think?
Because DNA will act as a genetic material. So genetic material changes, and the entire functioning of the organism is supposed to change.
Yeah.
But RNA will be transferring to protein, it will be further RNA and then protein, and protein will be applicable for some specific processes of the body.
Okay. So what happens if you're producing wrong protein? What will happen? There are machinery which can chop it up and throw it away. Okay, but if you make mistakes in DNA, that's it. You screwed up. You will get a cancer. So the nature also optimizes this way. So you understand, you can create wrong protein. It's not a problem. Then there are other enzymes which can cut it, ubiquitin, and you can use it again. But that's not. But DNA is a much more important thing. So nature also somehow optimizes what is the tolerable level here, right? The tolerable level is not the same depending in the same organism, depending where you're using this. Okay, and proteins is okay to make mistake, right? You can, yeah. So suppose you do this your bread and maybe one is not doing well. It's not a problem. You can throw it away, do it another one, right? But if you completely destroy the flour, right, then you can do nothing. That's an analogy.
Does the randomness affect this precision also as well?
It is, of course, right. So you see there is always a range a little bit, right? There is always a range, and there is always a range. So there is always this stochasticity, randomness, but overall, the outcome is still okay. Outcome is still more or less more precise. Okay. Yeah. So. Any more questions? Again, this was an example of first passage calculation, but you could do some other calculation. And the reason I would like to repeat first passage calculation is convenient here, because you really have a process with two possible outcomes, right one versus wrong one. And the first passage is convenient for this. Because splitting probability tells the probability to be right versus probability to be wrong. Any questions?
If not, let's then thank the speakers.
Thank you.
What is the plan?